Welcome to my series 100 Scientists Who Influenced the World, where I've taken a diverse mix of people and broken them down into 10 different categories. There are some famous scientists as well as some lesser known ones, but all of these people have had an influence on the world. Life on Earth is incredibly diverse and biologists attempt to understand living organisms. From early ideas of evolution to modern genetics, biologists have shaped how we see life today. So who are some of these brilliant biologists? Gregor Mendel. Gregor Mendel was born in 1822 in Austria to a struggling family. A local priest recognised Mendel's academic abilities at the age of 11 and suggested that he be sent to school where he studied mathematics, physics and philosophy. As his father's only son, Mendel was expected to take over the running of the family farm. However, when he was 21, he decided to join the Augustinian order, becoming a monk and freeing himself of those early struggles. Mendel grew more interested in science than religion and decided to start experimenting with pea plants to try and understand how traits of the plants could be passed on from one generation to the next. For years, people had been crossbreeding animals and seeing a clear mix, but the reason why was not understood. Mendel focused on 7 traits of pea plants: plant height, flower position, flower colour, pea shape, pea colour, pod shape and pod colour. He grew around 30,000 pea plants during his experimentation, crossing plants of different traits with each other, and he noticed some traits skip a generation before appearing again. To describe this, Mendel devised the terms dominant and recessive, terms we still use today to describe genes. He recognised that traits were controlled by a pair of factors, one from each parent plant. Before Mendel's work, nobody understood how inherited traits are passed on, but his experiments showed that they followed particular laws, now called Mendel's laws, and his work laid the foundation for further studies on evolution and genetics. Rachel Carson. Rachel Carson was born in 1907 in the United States, and early on she developed an interest in the natural world. Initially, Carson entered college with the ambition of becoming a writer, but soon changed her mind and switched her studies from English to biology, achieving bachelor's and master's degrees, and later undertook more studies in marine biology. Becoming a scientist did not stop Carson from being a writer, and her first book, Under the Sea Wind, was widely praised, and her second book, The Sea Around Us, became a national bestseller, won a National Book Award, and was translated into 30 languages. Carson became concerned about the widespread use of chemical pesticides on farms designed to protect crops by killing pests, but research showed their harmful chemicals were affecting food chains and fragile ecosystems. To raise the alarm, Carson wrote the book Silent Spring, with a title based on birds dying from exposure to chemical pesticides and the chances there could be a spring without birdsong. Carson was threatened with lawsuits by the chemical industry and was accused of distorting information and playing on people's emotions. Despite this, she stood behind her warnings of the indiscriminate use of pesticides, and although she died before she saw any real change, she left behind some of the most influential environmental writing ever published. Nettie Stevens. Nettie Stevens was born in the United States in 1861, and following the death of her mother when Nettie was 2, she was raised by her father and his new wife. Her father was a carpenter and earned enough money to provide a good education for Nettie and her sister. Stevens achieved degrees in biology and became increasingly interested in histology, the study of the microscopic anatomy of cells, and completed a year of graduate work in physiology and focused her PhD studies on cytology, the study of cells. Stevens followed these studies by taking up teaching positions while continuing with her own research. By observing the chromosomes of insects, Stevens discovered that for some species, the chromosomes are different between the two sexes. At the time, it was not known what caused animals to be born male or female, but further study from Stevens researching mealworms uncovered the X and Y chromosomes contained within their cells, though they were not called X and Y at the time. By studying these chromosomes, Stevens identified that male mealworms have XY for their chromosomes and females have XX for their chromosomes. A mealworm egg will always receive an X chromosome from its mum, as those are the only chromosomes she has to give. However, the egg will either receive a Y chromosome from dad when fertilised or an X chromosome. If a mealworm received XX, it was female. If it received XY, then it was male. With this, Stevens solved a mystery that had been puzzling scientists for a long time, showing that the sex of an animal was based on genetics and not factors in the environment. Andreas Vesalius. Andreas Vesalius was born in 1514 in Brussels, which was then part of the Netherlands, but is now part of Belgium. Vesalius was born into a family of physicians and pharmacists, so it was of no surprise that he followed down the family path into medical science. While studying in Paris, Vesalius had the opportunity to dissect animals as well as human bodies. He dedicated a lot of time to studying human bones. When he was appointed a lecturer at the University of Padua, one of his duties was to deliver anatomical demonstrations. Knowing a thorough knowledge of anatomy would be required, Vesalius conducted all his own dissections. At this time, the work of Claudius Galen was still the authority on anatomy. However, Vesalius observed this was not based on dissection of the human body, as that was banned by the Romans at the time of Galen, so Vesalius set about writing his own textbook based on his work. Vesalius' landmark publication, The 7 Books on the Structure of the Human Body, was published in 1543, the most extensive and accurate description of human anatomy than anything that had come before it. As a result, anatomy became a scientific discipline in its own right, with far-reaching implications for all of biology. Antonie van Leeuwenhoek. Antonie van Leeuwenhoek was born in 1632 in the Netherlands. Life had a sad start for van Leeuwenhoek. When he was 5, his father died. His mother remarried, but then when van Leeuwenhoek was 10, his stepfather died. Van Leeuwenhoek had very little formal education before being sent to live with his uncle. He had various jobs growing up, including a bookmaker's apprentice, ran a draper's shop, a chamberlain, a land surveyor and a wine gauger. While running his draper's shop, van Leeuwenhoek wanted to see the quality of his thread clearer than the magnifying lenses of the day would allow. He therefore made his own lenses and kept his method a secret, though a friend did encourage him to send letters about his observations to the Royal Society. Van Leeuwenhoek did so, starting with his microscopic observations of mould, bees and lice, and this began a series of letters back and forth right up until van Leeuwenhoek's death in 1723, totalling 190 letters the scientist had sent. One day, van Leeuwenhoek used his microscope to look at some pond water and was amazed to find microscopic life, the first observed bacteria. The Royal Society were not convinced, as there was no evidence for such life. However, this had many scientists verify results of the experiment, and van Leeuwenhoek's results were fully acknowledged, and he went on to make many more observations, including blood cells. Van Leeuwenhoek holds a very important place in the history of microbiology as the first to look and have the power of seeing the microscopic world around us. Barbara McClintock. Barbara McClintock was born in 1902 in the United States to a doctor father and an artist mother. When she attended Cornell University, she became interested in botany and plant genetics, eventually received a bachelor's degree in agriculture and a master's and PhD in botany. While studying the movement of genes within corn, McClintock identified that chromosomes are the foundation of genetics and that some genes have the ability to move within or even between chromosomes from one generation to the next. She referred to this as jumping genes and additionally found that some of these moving elements could turn genes on or off, which accounted for the variation seen in corn plants. McClintock's findings were initially dismissed, with people describing her ideas as radical. However, in the 1960s, scientists discovered that McClintock was correct. Recognition and awards came flooding in, including the National Medal in Science in 1970 and the Nobel Prize in Physiology or Medicine in 1983. McClintock's discoveries changed our understanding of genetics forever. Scientists learned that they could engineer genes, making them perform specific functions to produce desired traits, and it helped improve our understanding of the genetic basis of diseases and how to develop new treatments. Carl Linnaeus. Carl Linnaeus was born in 1707 in Sweden, and he was interested in plants from an early age. He attended university to study medicine, however, due to his poor financial situation, he could only attend a few lectures. Linnaeus had to subsidise his studies by teaching botany in the gardens of the university. At the time, if you wanted to open a medical practice in Sweden, students had to complete their degrees abroad. Linnaeus found someone to finance his studies in the Netherlands, and when he returned, he sought someone to finance the publication of his many manuscripts. He was immediately successful, and The System of Nature was published. This work laid a new way of classifying plants and animals, developing a hierarchical structure, replacing existing biological classification systems. This new system replaced chaos with order, creating a cornerstone for other biologists who further studied this field, and it is still the backbone of classification within the life sciences today. Sir Alexander Fleming. Alexander Fleming was born in 1881 in Scotland, and in 1895 he moved to England. He studied medicine at St Mary's Hospital in London and then began working there as a researcher, looking for antibacterial substances that would be non-toxic to humans. During World War I, Fleming served with distinction in the British Army Medical Corps and was increasingly upset by the number of soldiers dying from infections. In 1918, he resumed his research at St Mary's Hospital. As part of his research, Fleming identified and isolated lysozyme, an enzyme found in certain animal tissues and secretions, such as tears and saliva, which plays an important role in providing protection against bacteria, viruses and fungi. One of the uses of this enzyme presently is to aid in the preservation of foods. While working with the Staphylococcus bacteria in 1928, Fleming noticed there was a patch of mould which had got into one of his samples, but there was no bacterium around the mould. Upon investigating, he discovered there was a substance within the mould which prevented the growth of bacteria. By complete accident, Fleming had stumbled across an antibacterial substance that would change the world. He named it penicillin. Fleming lacked the necessary skills to isolate and identify the active part of the bacteria. However, he did get together enough penicillin to start treating skin and eye infections. It wasn't until 1939, because of World War II, when there became a pressing need for new antibacterial medicines, that Ernst Boris Chain and Howard Florey expanded on Fleming's work and were able to isolate, purify, test and then produce penicillin in high quantities. Fleming, Chain and Florey all shared the Nobel Prize for Physiology or Medicine in 1945, and although this section is focused on Fleming, we should not forget the great work of Chain and Florey, who helped bring penicillin to the world. Edward Jenner. Edward Jenner was born in 1749 in England and grew up in the countryside. When he was 13, Jenner was sent to be an apprentice of a nearby surgeon, where he learned about medical and surgical practice, before moving to London to work under one of the most prominent surgeons of the time. During the 18th century, smallpox was widespread, and occasional outbreaks of particularly high intensity resulted in a very high death rate. It is estimated that smallpox killed somewhere between 10 to 20% of the British population, higher in areas of greater population and lower for those in more rural areas. Jenner had heard of people in China deliberately infecting people with smallpox to help their body build their immunity to the virus. Unfortunately, the strength of the virus being passed on could not be controlled, and some people could end up seriously ill. It also meant that person could also become a spreader of the illness. Jenner discovered that if someone has been infected with cowpox, which doesn't have a big effect on humans, they become immune to smallpox. Jenner decided to expose people to a shot of the less impactful cowpox to help give them immunity. The first smallpox vaccine was given to an 8-year-old boy and proved to be successful. However, initial uptake of the vaccine was slow, but it rapidly proved its value, and Jenner was active in promoting it while seeking no financial gain. Jenner was recognised for his work in his lifetime, receiving multiple awards. Jenner remained a champion of vaccines throughout his life, and smallpox has pretty much been eradicated from most countries. Jennifer Doudna. Jennifer Doudna was born in 1964 in the United States. Growing up in Hawaii, Doudna was interested in the beauty of nature that she saw all around her, and she wanted to understand the biological mechanisms of life. Her parents also encouraged intellectual pursuit and enjoyed reading about science, so her home was filled with popular science books. Doudna's interest in science was also encouraged by her teachers in school, and she went on to achieve a bachelor's degree in biochemistry and a PhD in biological chemistry and molecular pharmacology before going on to work as a researcher at Massachusetts General Hospital and at different universities. Doudna had been researching RNA, a single strand of genetic information similar to DNA, when she came to the attention of a colleague who was seeking people to conduct research with CRISPR, a natural segment of genetic code found in most bacteria. Doudna and her colleagues discovered that by using a protein called Cas9 found in the bacteria Streptococcus, they could reduce the time and work needed to edit genomic DNA. Doudna, along with Emmanuel Charpentier, showed for the first time that they could use different RNA to program cuts and edit in different DNAs. For this discovery, the two ladies were jointly awarded the Nobel Prize in Chemistry. CRISPR technology has been further developed by other research groups for a range of applications, including treatments for diseases such as sickle cell anemia, cystic fibrosis, Huntington's disease and HIV, though Doudna and many other leading biologists are wary of CRISPR and how they believe it should be used. Either way, this discovery is having a huge impact in the world of biology. Thank you for watching this video in my series 100 Scientists Who Influenced the World. The majority of the information for this video came from the books 100 Scientists Who Influenced the World and the Britannica Guide to the 100 Most Influential Scientists, as well as some other online sources. If you liked this video, please hit the like and subscribe buttons to stay up to date on all future content, and be sure to check out my other videos such as my STEM demonstration and explanation videos, my STEM career interviews and my 10 Things You Should Know About series. This has been STEM with Mr. N's 100 Scientists Who Influenced the World. Brilliant Biologists.